Hey team, here we are for chapter 25. It's all about the history of life on Earth. This is going to be the condensed version of the history of life on Earth. Obviously, we could take an entire semester to discuss how life has changed over the past billions of years here on Earth. We're going to look at a very shortened version of that, basically from the very beginnings of the formation of the Earth and early life all the way up to basically the present day in our current epoch of time. So this is a history of life on Earth. Chapter 25 is basically about all the large-scale evolutionary changes that have occurred, the rise of various new groups of living creatures such as flowering plants or land-based mammals and the like. We're going to find out that the vast majority of time for history of life on Earth has been without most of the creatures that we see today. So for many millions of years, most of the living creatures on the planet were very simple compared to some of the creatures we see today. They were for the most part prokaryotic, and then there were some very simple unicellular eukaryotic, and then we finally get to multicellular, and then there's quite some time before we get to the animals and plants that we're familiar with. And of course humans have only been around for a very brief span of time in comparison to the geologic timescale that we're going to look at. So this is a brief, condensed version of the history of life on Earth. Let's start with a brief example of how things have changed so dramatically in the billions of years of geologic time that have occurred on planet Earth. Imagine some of the early explorers that visited Antarctica, what we now know as Antarctica. So we're talking about the South Pole, the bottom of the planet, perhaps one of the most treacherous and unforgiving places on Earth. Very little actually lives there, specifically on the land. There are only a few organisms that call Antarctica home as far as creatures that live strictly on the land. There are, of course, various mammals and other fish that live in the waters and use the land to some degree, but not a whole lot of living creatures there on Antarctica. Very cold, very windswept, not very hospitable to life. So imagine some of the early human explorers to Antarctica trying to reach the South Pole, things of that nature. What do you possibly think crossed their mind when they began to discover fossils there in Antarctica, fossils of amazing creatures that were no longer in existence, and evidence that Antarctica not only didn't always look the way it looks now, but the fact that Antarctica was at one time an area that was warm and almost tropical and covered with oceans and water and lakes and forests, quite different from what we see today. So the past organisms that lived on our planet were very different for the most part from those that are alive now, from the ones that we see around us today. And the fossil record is a great way of seeing some of this macroevolutionary changes that have occurred over these vast amounts of time. So we're talking major categorical changes like the emergence of terrestrial vertebrates, the impact of mass extinctions, which there have been numerous, the origin of flight in birds, major things that have shaped the biology of our planet. Remember, macroevolution is the broad pattern of evolution above the species level, so we're talking about major groups of living creatures and how those have come to be. Here's an artistic illustration of perhaps what Antarctica looked like at a certain time, approximately 190 million years ago. We have fossil evidence of this particular type of dinosaur that lived in the area, and we have fossil evidence of the various plants and the way that the land looked. And it's difficult to imagine that this at one time was the land that is now Antarctica. Of course, we're going to find out it wasn't in its current position on the globe, and there were quite a few differences between the land that we now call Antarctica and what you see in the drawing here. But these creatures did live on that same piece of land, but obviously things were quite a bit different 190 million years ago. Here's the skull of that same dinosaur that was shown in the drawing there. This is Cryolophosaurus. Boy, I love to say these dinosaur names. Cryolophosaurus, a skull of that particular animal, and you can see that interesting crest on the top of its head. And there's a lot of information that we can learn from the fossils of these creatures, and we'll talk more about the fossil record and what evidence it shows for the changes that have occurred here on our planet. We're going to start right here with concept 25.1. It says the conditions on early Earth made the origin of life possible. And of course, we don't know exactly how life originated here on planet Earth. The origin of life is going to be the most speculative topic of this entire unit because we don't have any direct fossil evidence of that particular episode or moment in time. But you need to be sure and understand that observations and experiments in chemistry and geology and physics have led scientists to propose a scenario that we're going to examine here, and we use the tools of science to answer what we can, and we refine as we learn more. It may be the case that we never confidently or totally understand how life began on our planet, but we certainly have evidence that points toward a possible scenario, and that's what we're going to look at today, is what is the various pieces of scientific evidence that kind of support certain hypotheses. We don't know for sure, but in reality we know nothing for sure in science. In science you are very hesitant to say this is 100% or this is incontrovertible without a doubt. Science is all about the possibility and basically the confidence you feel in your evidence. The greater amount of evidence you have and the greater the number of lines of evidence, the stronger you feel in your hypothesis being correct or it being an accurate reflection of reality. So some people think that's a negative about science or a weakness of science, when in reality it's quite the opposite. The fact that we do change our thoughts over time, that we perfect our models and make them stronger as we learn more, in my opinion that is a strength of science, the willingness and ability to learn and add new information as those facts present themselves. So let's take a look at one possible scenario, and again this is a scenario that is deemed most scientifically likely simply because of the evidence that we have that supports this particular idea. Chemical and physical processes on early Earth may have produced very simple cells through a sequence of stages. First stage would be what we call abiotic synthesis of small organic molecules, then the joining of these small molecules into macromolecules, the packaging of them into what we would call protocells, or kind of the basic beginnings of what would eventually be called a cell or defined as a cell, and then the origin of self-replicating molecules. None of this took place in a speedy manner. This was probably over the course of millions, perhaps billions of years, and we will take a look at the evidence that kind of helps support this idea. And although this is speculative, the scenario does lead to predictions that can be tested in the laboratory. So this is not just a wild idea that was thought up in someone's dream one night. It is a thought process that does lead to certain testable predictions, and if those predictions are borne out by the evidence, that gives you a bit more confidence in the scenario. So let's take a look at some of this evidence and talk about how perhaps life began on Earth. Scientific evidence supports the fact that the Earth was formed about 4.6 billion years ago, along with the rest of our solar system. And early in the solar system, it would have been a pretty wild place. You would have had the new formation of planets going on. You would have had a huge amount of debris that hadn't yet accumulated or accreted into the planets we see now. The solar system, in comparison now, is a pretty clean place. Yes, there are meteoroids and comets and asteroids, but not nearly as much debris as there would have been in the early formation of the solar system. Bombardment of Earth by rocks and ice likely vaporized water and prevented seas from forming before about 4.2 to 3.9 billion years ago. And the early atmosphere of the Earth likely contained a large amount of water vapor and various chemicals released by volcanic eruptions. You can imagine the Earth was quite active volcanically as far as the land formation and before we had the cooling of the crust and the formation of the solid crust, there would have been enormous amounts of volcanic activity and geologic activity releasing nitrogen and nitrogen oxides and carbon dioxide and other gases into the air. In the 1920s, there were two scientists, Operan and Haldane, that hypothesized that the early atmosphere of Earth was likely a reducing environment. Remember, from chemistry, reducing means electron adding environment, in which organic compounds could have formed from simpler molecules. And the energy for such a reaction, remember we have to have this activation energy to kick off any chemical reaction. They said the energy from this reaction likely came from lightning or intense UV radiation prior to the formation of our atmosphere and some of the radiation blocking components of our atmosphere that we have today. Haldane suggested that the early oceans were likely to be a solution of organic molecules, and he called this a primitive soup from which life arose. And all of the scientific evidence that we have shows that life first began in these watery environments within the ocean. And as a matter of fact, for many, many, many years, life existed only in the water, in the oceans, before it came onto the land. In 1953, if you'll recall, we talked about this experiment last semester. Stanley Miller and Harold Urey conducted lab experiments that showed that the abiotic synthesis of organic molecules in a reducing atmosphere is possible. If you'll recall, they had this primitive soup, this liquid that contained certain chemicals, and then they passed electricity through this atmosphere, through this material, and abiotically, meaning without the help of living creatures, pre-existing living creatures, there were organic molecules that were synthesized through this process. So that was something that we kind of talked about last semester when we were discussing organic molecules in chemistry, and that helps to clarify or to give some strength to this hypothesis. Now, of course, I said this is not a cut and dried done deal. There are some scientists who believe that there's not enough evidence to show that the early atmosphere was reducing. In fact, they believe that perhaps the atmosphere was perhaps more neutral. It wasn't oxidizing or reducing. So instead of forming in the atmosphere, the first organic compounds may have been synthesized in small local areas near volcanoes or deep sea vents, where the atmosphere would be reducing and would be conducive to this formation of these organic molecules. The Miller-Urey type experiments demonstrate that organic molecules could have formed with various possible atmospheres. However, these types of experiments have been repeated numerous times in numerous ways with a variety of possible atmospheres, and inevitably organic molecules are able to be formed through this abiotic process in a variety of possible environments and possible early Earth atmospheres. Throughout the PowerPoint here, you're going to see little sections that have video clips. I didn't embed these necessarily in the PowerPoint, or you won't be able to see them just in the vodcast, but if you have the PowerPoint itself, you should be able to click and play those. And if not, I did upload all of the various videos to the notes folder for this chapter. So if you wanted to see some of these first videos talk about tube worms, and they show also some hydrothermal vents, which would be at the bottom of the ocean, where you have basically volcanic activity at the bottom of the ocean, unique environments where even today we find a variety of unique creatures that you don't hardly find anywhere else, creatures that go through their metabolic process differently than many other creatures that we find on Earth. These could have been the nurseries for some of the first life on our planet. And then here we have another example of the Miller experiment. In addition to his classic 1953 experiment, Miller also conducted an experiment which simulated a volcanic eruption, and in a 2008 reanalysis of those results, researchers found far more amino acids were produced under these simulated volcanic conditions than were produced in his original experiments. So again, that would kind of support this idea that perhaps there were locations on the planet, hydrothermal vents or near volcanic activity, that provided the environment that would be conducive to producing these initial molecules that would go on to produce living creatures. Another possibility for the formation of these first organic molecules is the fact that there are many organic molecules, amino acids and the like, that have been found in meteorites that have fallen to Earth. There's a type of meteorite called a carbonaceous chondrite, and they are 1 to 2% carbon compounds by mass, and within some of these meteorites you can find all sorts of various amino acids and organic molecules. There's a particular meteorite called the Murchison meteorite, a fragment of it, about a 4.5 billion year old chondrite. So this meteorite is about the age of the Earth itself. It fell to Australia in 1969, and it contained more than 80 amino acids, and some of them were in fairly large amounts. These amino acids, you might say, well, how do we know those amino acids came from the meteorite? How do we know they weren't basically contaminants from Earth when the meteorite fell to Earth? Well, the main reason is because inside the meteorites, the amino acids consist of an equal mix of two separate isomers called D and L isomers. You remember we talked about isomers back in chapter 4, where here on Earth, organisms make and use only the L isomers with only a few rare exceptions. So the fact that the isomers inside the meteorite were basically a 50-50 mix of D and L tells you that it didn't likely come from Earth. And recent studies have shown that this Murchison meteorite that we're talking about not only did it contain amino acids, but it also contained other organic molecules such as lipids and simple sugars and nitrogenous bases like uracil, some of the same materials, one of the 4 nitrogenous bases that makes up our DNA. So there is possible evidence that some of the material that eventually was needed to create life could have come from outside of our Earth at one point. The formation of these amino acids isn't an important step possibly in early life, but amino acids alone aren't enough. We have to string these amino acids together. We have to create macromolecules, and it's macromolecules that then combine to form the living parts of creatures that we see today. So where do these macromolecules come from? How can we possibly see the abiotic synthesis of these macromolecules? Well, there was a 2009 study that demonstrated a key step where the abiotic synthesis of RNA monomers was created, and this occurred spontaneously from some simple precursor molecules, some simple building block molecules. So we found that in some cases in the lab we can show experiments where this can happen on its own, where these macromolecules form through a process of a combination of some of the more simple precursor molecules. And then there was another experiment that your book talks about where it says, in addition to that, by dripping solutions of amino acids or RNA nucleotides onto hot sand, clay, or rock, researchers have produced polymers of these molecules. Remember, polymers are the long chains, and they are the beginnings of some of these macromolecules that make up living creatures. The polymers form spontaneously without the help of enzymes or ribosomes. So for a long time it was thought, how could these macromolecules be created without the help of some of the various enzymes or ribosomes that our cells use, living creatures use? But there are processes where this happens by itself, spontaneously, without the need for those parts. How did we move from these macromolecules to what would eventually become a cell, something that we would define as living? Well, let's start with what we might call a protocell, not necessarily something we would define as a cell today as we know it, but the beginnings of potentially a cell. For it to be alive, it has to reproduce, it has to replicate, and it has to have metabolism. In other words, it has to keep its internal environment separate and apart and perhaps different from the external environment. Those are key properties of what we mean when we say something is alive, and they may have kind of appeared together at the same time or within the same time frame. We'd call these early possible becoming cells protocells, and they may have been fluid-filled vesicles, and a vesicle is simply a bubble, a membrane-like structure which has an environment on the inside different from the environment on the outside. For example, when you drop cooking oil into water, it forms vesicles, little round bubbles where the interior is separate and different from the exterior. So that would be kind of a basic description of a vesicle. Recent experiments have shown that these abiotically produced vesicles can exhibit certain properties of life. In a way, a simple type of reproduction and metabolism. In other words, they have a way of keeping the internal separate from the external and a way of basically subdividing or reproducing themselves. And again, they mentioned, for example, in water, lipids and other organic molecules spontaneously form vesicles with a lipid bilayer. If you remember, our cells, the cell membrane is made up of a lipid bilayer, and this kind of mirrors what can happen with these protocells that we're talking about. In addition, scientists have found that the addition or the adding of clay, very, very fine particles, usually say something produced from the weathering of volcanic ash, and we already talked about how active volcanic activity was in early Earth. The addition of this fine clay material provides a substrate for these macromolecules to kind of gather on, and it increases the rate of vesicle formation as well. So now you have this substrate, this material basically that's kind of almost like chemical Velcro, where you have these macromolecules collect and become concentrated and can interact with one another. So in a way, the clay material would act not only as a substrate, but almost like as an enzyme. It would assist in the interactivity between these macromolecules. In your textbook they have some good illustrations and pictures. So here it's talking about the self-assembly of these vesicles, how they can form, and it talks about how when you only have the little precursor molecules, the formation of vesicles is fairly slow over this time period. But if you add a little bit of this clay material and give it something to attach to, a substrate, the formation of these vesicles increases exponentially at first. It jumps up dramatically before it starts to level out. Then you see some vesicles here where they talk about basically reproduction, subdividing, and then here we have the absorption of an RNA molecule within a vesicle, a non-living vesicle, a protocell you might consider it. One stumbling block for the early scientists studying this idea of the beginnings of life on Earth was, for DNA to replicate, you need all these other materials. You need DNA to make DNA, and it was kind of a chicken and egg problem, and it was difficult to figure out how did this process work. But the more we've studied this and the more evidence we've looked at, it seems like DNA was not where we first began. It seems like RNA was perhaps the first genetic material in these early cells. RNA, which we already know plays a central role in protein synthesis, it can also do a number of other jobs. It can carry out some enzyme-like functions, some catalyst-type functions. And there are RNA molecules that we call ribozymes. Ribozymes have been found to help along, speed up, catalyze a lot of different reactions. So ribozymes can make complementary copies of short stretches of RNA. So RNA, it turns out, can be self-replicating in a way, and then it can also act as an enzyme. So it can kind of serve a dual role. Now in our modern cells, it doesn't really serve that same dual role. We have enzymes and we have RNA, and then we have DNA, which is now our master genetic copy. But in early cells, RNA may have basically worn multiple hats and done multiple jobs, and that looks like it's the case based on the evidence we now have. We've already discussed a little bit about how RNA is different from DNA. DNA tends to be a nice, stable, uniform double helix shape, but the single-stranded RNA takes on assumes a variety of shapes based on its nucleotide sequence. So a different nucleotide sequence will cause it to form into a slightly different three-dimensional shape, a lot like the proteins that we discussed, where the sequence along the amino acids of the proteins cause it to take a different shape. Well, the same thing occurs in RNA, except we're not talking about amino acids, we're talking about nucleotides, but the different sequence of nucleotides cause the RNA to take a different shape, and this could provide differences. And remember, when we talked about differences, suddenly you have the ability for natural selection to begin working on these differences. And RNA molecules that were more stable or perhaps replicated more quickly, they would have been favored by natural selection in these early cells or protocells, and they would have left what we might call more descendants of RNA molecules. So this early genetic material might have formed what you might call an RNA world, and it may have been a world before the eventual formation and then the use of DNA that the living creatures now have today. These vesicles with RNA capable of replication, they would have been these protocells, the beginning stages of what we would now call a cell. And then later on, RNA could serve as a template for DNA, which turns out to be a much more stable and robust genetic material. And then obviously that would be favored by natural selection, and so DNA basically became the standard, or the gold standard, to the currency of genetic information from one living creature to the next. After DNA appeared, RNA perhaps began to take on more of what we consider its present-day role as a regulator or kind of an intermediary in the translation of genes. So in the taking of the information from the DNA and passing that information to the various protein synthesis materials such as the ribosomes. So now, once we have these protocells and we have the ability to reproduce and we have metabolism and we have differences within these cells that can be acted on by natural selection, you have the stage kind of set for the blossoming of various life forms. Now, for a long time, we're going to be talking about only single-celled creatures, even before we get to any sort of multicellular creatures, but we have the beginnings of the process. Now, we don't have fossil evidence of these very early stages, but we do have fossil evidence that picks up early on, and that's where we'll go from here.